So hello, everybody. Thank you for joining us. I'm Sharon Husterly. I'm the Senior Director of Research at Parent Project Muscular Dystrophy. And as many of you know who've been on other webinars, we've done a series of these webinars on topics of interest to the community. And today we are very pleased to have joining us Chris Garabedian, who is the Chief Executive Officer at AVI BioPharma. So Chris is going to give us some insight into AVI's plans going forward, and I will let him tell you more about what he's going to discuss. But first, I'll just say that, the presentation today will be relatively short. We're saving a lot of time for questions. And if you have questions, everyone on the phone is on mute. However, you should have access to the chat box on your screen if you're also logged into the webinar. You can type questions at any time during the presentation. If for any reason we don't get to a question, we will try to answer it offline. Or if you feel like it got buried, you can try repeating it, but hopefully we won't need to do that. So we are putting aside a significant amount of time for questions. Now, with that being said, I think that takes care of all the housekeeping. I will go ahead and turn this over to Chris.
Okay. Hi, Sharon, and thank you for the opportunities to use PPMD as a way to communicate to the Duchenne community, and particularly parents of Duchenne patients and those living with Duchenne. Pat Furlong has been a great resource for me, and she's a great link to the Duchenne community, and I can always count on her to help me keep a pulse on the discussions among parents, especially as it relates to AVI's efforts with our DMD clinical program. She recently informed me that the community is excited and anxious about the upcoming trial and haven't heard from AVI directly in a while, and offered this webinar as a forum to provide an update to answer any questions. Let me remind everyone that no new information will be shared on this call that has not been shared in open public communications previously, whether that be through investment conferences, press releases, SEC disclosures, or through our quarterly earnings conference calls. Many of you have seen or heard me present many of the slides that you'll see today, but I will be speaking about them in a different context, specifically what information we have collected in our Phase 2b study and the types of information that you can expect to see coming out of this study. Let me be very clear that we have not unblinded this study and are not aware of the trial results at this point. We have not seen the muscle biopsies, neither I nor Ed Kay, our Chief Medical Officer, nor anyone at AVI, and we will not see them until they are delivered with the rest of the unblinded data sets. Please do not ask any questions about the results, as we do not have anything to share at this time. First, I'd like to start off by highlighting a recent event that we announced, which is that we had a late-breaker abstract accepted at the American Academy of Neurology meeting that is to take place in New Orleans from April 21st through the 28th. The lead author on the abstract was Jerry Mendel, our principal investigator of our Phase 2b study, and it was accepted as part of their emerging science session, which was formerly called late-breaking science. It is in a format called Data Blitz, which is a series of 15 3-minute, 3-slide presentations followed by a lengthier poster presentation, which allows a poster with more data and discussion following the oral Data Blitz portion of the session. We will likely be sharing top-line results in a press release format prior to the American Academy of Neurology meeting, but the fuller top-line results will be presented at the meeting. So I'd like to turn to the slides, and I'm going to skip over a couple of the slides that many of you have seen me present previously. And Sharon, can I ask you, do I have control of the slides, or do I need to tell PPMD which slide I'd like to refer to?
You should.
Do it.
Yeah.
Okay. So, Danielle, if you can move to slide 5, which highlights the muscular dystrophy mechanism.
Sure.
Okay. Many of you understand that our goal of the treatment that is in the Phase 2b study is to produce dystrophin in these boys and patients who don't have the ability to produce dystrophin or simply get revertant fibers. So we know what a muscle tissue in a Duchenne patient looks like. It looks like the bottom cell here that's essentially black when you do immunohistochemistry staining. And a healthy muscle tissue looks like the cell on the top. And we will be getting pre-treatment biopsies from all of the patients in our study, and we will have post-treatment biopsies in those patients. We don't expect that we'll see muscle looking like a healthy individual, but we hope to have something closer to seeing a lot of green throughout the muscle that's definitely different than what we see in the pre-treatment biopsies, where there's essentially no discernible green staining that you see. If you can go to slide 6, which is a description of the dystrophin gene. Many of you have seen this. I won't spend too much time, but our mechanism of action is really trying to restore the reading frame so that the dystrophin gene can translate to the dystrophin protein. Duchenne, on the right here, you see is marked by an out-of-frame deletion. And by targeting a specific exon across the gene, it allows us to turn an out-of-frame deletion into in-frame by silencing or skipping over a specific exon, depending on the mutation or set of mutations in the patients we're trying to treat. The Becker's muscular dystrophy genotype is characterized by in-frame, where Becker's patients can produce more dystrophin and sometimes markedly more dystrophin than a Duchenne patient, and they tend to have a much milder disease and a different natural history. So effectively, we would like to achieve a clinical outcome that's more akin or reflective of the milder disease of Becker's. So that's the goal. And slide 7, I'll just briefly highlight as well, Danielle, is just an example of our exon 51 skipping drug. This is a patient with a deletion at 4950. By skipping over exon 51 with our technology, we can restore the reading frame and produce the dystrophin protein. This was the proof of concept we were trying to prove in our previous UK study that was published in the Lancet, slide 8, this past summer. Any of you could have accessed this. Was published last summer to give a more detailed highlighting of the results from our phase 2B. In slide 9, we show an example of a patient in our study where we were able to show the production of dystrophin. This is a patient pre-treatment and post-treatment biopsy in the lower right-hand panels. In the upper right-hand panel, we show evidence of Western blot protein expression, and on the upper left-hand panel, we show evidence of our mechanism of action via exon skipping through RT-PCR. We are going to be looking at all of these measures in our phase 2B study, both baseline, pre-treatment, and post-treatment. And we also have the benefit of having a placebo group to compare these results to, of what would happen, compared to a placebo untreated population. Slide 10 just highlights that we showed every patient in our top 2 dose cohorts, the 10 milligram per kilogram and 20 milligram per kilogram cohorts, showed novel dystrophin production. And we are now looking at higher doses in our phase 2B study, 30 milligrams per kilogram and 50 milligrams per kilogram. And we will be capturing data from our 30 milligrams over 24 weeks, which is, again, a longer duration of therapy, while we're looking at 50 milligrams at 12 weeks to compare it to these lower dose cohorts. Slide 11 is something else that we're going to be capturing in our phase 2B, which is the lymphocyte counts or inflammatory infiltrate in the muscle biopsy. Again, we're looking at pre and post effects of the treatment, and we did see a very nice dose-dependent relationship in our previous study, where at the higher doses we were able to show a robust reduction of these T-cell counts in the muscle tissue, CD3, CD4, and CDL, which we think is a marker of the creation of healthier muscle tissue. Most of the rest of my comments are going to focus on the study design of our phase 2B and how we designed it, the inclusion criteria, the endpoints that we're capturing, and also a little bit about the rollover extension study. If we go to slide 12, Danielle, this highlights the 24-week study 201, which is the placebo randomized blinded portion of the study that we've now rolled over. And these are the results that we're going to be sharing in top-line form in the month of April. As you can see, as I described earlier...
Can I interrupt you just for a second?
Sure, sure.
We've had a couple of questions, and I think I would like to know as well, when you say top-line, what exactly do you mean by that? Yeah,
sure, and I plan to go into more detail. The dystrophin data, which is our primary endpoint, which we defined as dystrophin-positive fibers as a % of normal, looking at the pre- and post-treatment cohorts. That is something we intend to share as part of our top-line results. That is the primary endpoint. We have captured a lot of other information and data, and again, we don't have the benefit right now of looking at that data and understanding it. And so we know that it will be a more limited data set that we put in the press release and a larger top-line data set at the American Academy of Neurology. We have to be very careful. When we announced our acceptance at the American Academy of Neurology, they made a couple phone calls saying that that we better not share too much information or we would risk having the presentation pulled. And so they understand, as a small publicly traded biotechnology company, that we can't sit on the data if we have it in hand. And so we do have to reveal the material top-line information, which we know will be dystrophin. We will have to make a judgment of how much more information we can share in a press release without compromising our ability to present the data at AAN. That's an important meeting because it's peer-reviewed. We do want to have a poster presentation at that meeting that we can talk about and refer to, throughout the year. We also know that there's a lot of people who attend that meeting to write up and report on the importance of data being presented at that meeting. So that is an important meeting for us to retain that late breaker emerging science oral presentation slot and poster review, and we don't want to compromise that by sharing too much information in the press release. So we will have to make a determination of the material elements that are required for our press release disclosure, but we're not a priori describing what is or is not going to be in that press release. But again, the primary endpoint of dystrophin is something that you are likely to see in a press release.
Okay, and that primary endpoint is what you would call top-line data as opposed to other kinds of data.
Yeah,
the term top-line, I think, is unfamiliar to people.
Okay, yeah, top-line, it's look, this is a study where we're collecting a lot of information. This will be a very rich data set. Many companies, whether they're private biotechnology companies or large pharmaceutical companies, don't have the same disclosure requirements of a small publicly traded biotech company. Many times companies take weeks or months to sift through the data to understand what it's telling them before they report on anything related to a study like this. We have to quickly discern what we call top-line data that is most relevant and material for our investors, right, to understand what is material about this study. And so that's what I'm referring to by top-line. This is a data set that we believe is rich, meaning that there's many different types of analysis that could come out of it that will likely be reported on for weeks and months to come. At future scientific meetings, we can do many different types of analysis, but that takes a lot of time. That's not something we are able to do, nor would we want to make a quick summary judgment on such an extensive database. So when I refer to top line, it's the most salient, critical information needed for the investors and community to understand if this is a positive study and if it is something that they were expecting, whether good or bad in terms of outcomes. So I hope that clarifies that question on top line data, Sharon.
Yeah, great. Thank you.
All right. I think I'll go to let's stay with slide 12. I want to highlight a little more detail than what we've typically shared previously. All of this I have shared in other public forums, but given that we're close to a data release, I think it's important to understand the inclusion criteria that was used and the patients that were selected for this study. The most important thing in our mind in doing a study is to design it and to include patients where we would have the best chance to show a treatment effect. We know we're dealing with small patient numbers, and we know we're dealing with a relatively short duration of follow-up, relative to a chronic disease that is progressive over years before loss of ambulation. Given those two factors, we wanted to select patients where we would have the best chance with small numbers and a shorter duration of follow-up to show a treatment effect. And we did this by studying the literature, by learning from other companies that have done studies in Duchenne, and learning from how these patients and boys are progressing based on certain risk factors or demographic characteristics, etc. So we decided to include boys who were between the ages of 7 and 13 years of age. That was an inclusion criteria. We also learned about progression of disease based on the baseline scores of 6-minute walk tests. And so we narrowed the inclusion criteria to try to get a patient population that would be more likely to show a progression of disease over a 6-month or 24-week time frame. So we decided to include patients who had a baseline 6-minute walk test of between 200 and 400 meters walked on their 6-minute walk test. We also learned things from other studies where we wanted to make sure we had a relatively stable group going into the study. So, for example, we've learned that sometimes when patients are enrolled in a study, they start adhering to their other medications, their steroids, for example, where they may not be fully compliant. When they're enrolled in a study, all of a sudden they're being more compliant with their steroid use. And then all of a sudden you see them performing better because they're taking their steroid medication. We wanted to make sure that in the pre-screening stages of this, that in the patients that might have qualified for the study, that we were trying to get a cohort that we worked with, Jerry Mendel led this effort to make sure that all the patients he was pre-screening, following, et cetera, were on a stable steroid dose, so we didn't see this effect coming into the study. Another thing that we saw from other studies is that when you enroll patients for the first time and they're doing a battery of physical therapy tests that some of which they don't do in terms of their normal visits with their physicians, we saw learning effects, right? So if they do these tests for the first time in a study, the second time they do it or the third time, they get to know the test better and they perform better because of that learning effect. We wanted to try to minimize the learning effect, so we had patients go through these exercises prior to the baseline scores so that we could mitigate this learning effect and that might confound the data. So these were things we were very thoughtful about the patient selection and how they came into the study so that we could get the maximum amount of information and understand the treatment effect versus placebo. We also decided to look at a variety of endpoints because this is the first placebo-controlled study looking at exon skipping that will be disclosed. And when you're comparing against placebo, we really don't know what exon skipping is going to have the biggest impact on in terms of clinical outcomes. So we wanted to collect a variety of outcomes to better understand what an exon skipping drug effect could have, and what are the best measures and clinical outcome tests that would demonstrate a treatment effect in the most robust way. So we collected a variety of endpoints, and we decided to collect them frequently, every 4 weeks, so that we have a good area under the curve, rather than a couple data points which are hard to discern a trend. The other thing we did is, because with the small numbers we've evaluated here, we wanted to make sure that there wasn't the risk of a bad performance day. In other words, sometimes the boys are tired a certain day and don't want to perform the test, or there's an anomalous data point. So we decided at the baseline 12-week and 24 weeks on some of the key endpoints, we decided to retest and keep the boys overnight so we could capture 2 measures and try to mitigate some of the anomalous data that might confound or compromise the real treatment effect that we see. So I just wanted to highlight to the parents and other community members on the line that we were very thoughtful about this study design. We are very anxious as well, and hopeful that this study will inform us to the true effects of our drug, and most importantly, to help guide a design of a confirmatory or pivotal study that we would hope would move forward, and that we could start enrolling by the end of this year, with dosing starting in the first half of next year. Danielle, if you could go to slide 13. I just wanted to highlight that we have been very pleased with the safety profile of this drug to date, both in our previous UK study as well as in this study. The DSMB, our Drug Safety Monitoring Board, met in January to look at the 50 milligram biopsies as well as all of the lab results that had occurred, and gave it a clean bill of health to proceed. Again, we have not seen any serious adverse events that would cause concern across the overall population, so that would be regardless of whether they were on treatment or blinded placebo. Again, we're very encouraged by the safety profile to date. We continue to explore much higher doses in animal toxicity studies, and we have completed now dosing in primates out to 9 months at 320 milligrams per kilogram, and in mice at 900 milligrams per kilogram over 6 months, and we will have the results of that in the coming months as well. But to date, we have seen a very strong safety and side effect profile, and we think this is important in a population that may require drug therapy, chronically throughout their life. So we're very pleased with the safety profile. On slide 14, Danielle. The last thing I want to do is highlight that we have completed that 24-week blinded phase of the study, and now have rolled over every patient to open-label eteplirsen. And we did not have any interruption of dosing in those treated patients, and the placebo patients rolled over immediately. So in week 25, they were on open-label study drug. So now we have 12 patients on the drug that we're continuing to follow, 6 of them at 30 milligrams per kilogram and 6 at 50 milligrams per kilogram. We don't know what the best dose to bring forward in a pivotal study is at this time, so we wanted to continue to capture safety and efficacy at both of the dose cohorts moving forward. We also wanted to add a third biopsy, which we think could be of critical importance in our discussions and negotiations with the FDA. We thought long and hard about the requirement of these boys in the study to subject them to a third surgical biopsy. However, we do believe that 48 weeks of treatment and looking at the 48-week data of dystrophin will be of importance to the FDA. The other thing we will have is 24-week dystrophin data of the placebo patients that have rolled over. And while we staggered the biopsies in the 24-week stage, this will also allow us to compare more directly 30 milligrams versus 50 milligrams of dystrophin production. So we thought hard about this, but we thought in the end that third biopsy could be of critical importance to getting this drug approved and maybe even under an accelerated format, depending on what the clinical outcome data looks like. We may have that data by year end, but we haven't done the full protocol yet to understand when we would be able to have that data. The nice thing about the open label portion of the study is that we can continue to monitor the safety of this on an ongoing basis, and we have dialed in some efficacy measures in a continuous fashion as well, just not as frequently as we did in the 24-week stage. So with that, slide 15 just highlights the time frame of the data. Again, within the coming weeks, we expect to have that top-line data in a press release and more data to be shared at the American Academy of Neurology meeting. The time of that presentation is in the afternoon in New Orleans on Wednesday, April 25th. And that is in our press release, the more details around that. We also expect to have our long-term animal toxicology results sometime before the end of the second quarter, possibly in June. And again, all of that we will prepare in a briefing document to submit to the FDA and request an end of phase 2B meeting with the FDA that we expect to happen in the third quarter. This will be an important meeting to help guide our clinical program moving forward and to discuss what a pivotal or confirmatory trial design would look like and what's acceptable to the FDA. It's premature to talk about it, but I get a question often about the opportunity for accelerated approval filing. That will be dependent on how robust the data is and how much we're able to show a treatment effect with our 24-week study results. And it will also depend on the FDA discussions and what their feeling is about a early filing under accelerated approval NDA. The last slide I'll just highlight is slide 16, which is we've been fortunate to have two collaborations which will focus on our follow-up exon skipping drugs. We have a collaboration with two academic centers, Children's National Medical Center out of Washington, D.C., and Carolinas Medical Center out of Charlotte, North Carolina. These organizations have received funding from the DoD and the NIH, respectively, to explore an exon 45 drug with AVI's morpholino technology, which we are on our way to selecting a sequence and preparing to do the IND enabling work to get a drug, ideally with an open IND by the end of next year. It's hard to predict when trial would start, but the goal is to have an open IND by the end of 2013. The same is true with our exon 50 collaboration with NIH. This is their TREND program, the treatment of rare and neglected disease, or therapeutic for rare and neglected disease, where they help bridge the gap to do IND enabling work to get rare disease drugs into the clinic. So we were pleased to announce last year that we got an award to focus on an exon 50 drug. And again, the goal there is to have an open IND by the end of 2013 as well. So with that, Sharon, I hope that provides a nice update and overview to the DMD community, and I'm happy to open it up for Q&A. My website here that I'm looking at, it's very hard to discern all of the questions. I'll try to scroll through, but I'm happy to have you start by asking any questions that you know that the community would like to hear an answer on.
Okay, great. Chris, that was really comprehensive. I think that has helped answer a lot of questions, but of course we still have plenty coming in. So I think I'll start with the first question that I think is really important, and that is, what percent dystrophin do you think is going to be needed to get some functional efficacy? In other words, to improve muscle strength, or not necessarily to improve muscle strength, but to slow loss of muscle strength. Because you talk about how robust your ability to fast track will depend on how robust that data is. Do you have a target?
Yeah,
I should add.
Yeah, no. It's a great question, and it's one that I think the research community would like to better understand. I can tell you that what I would hope to see, and what I think is a worthy goal, is to see at these higher doses that we're testing that we start to approach or see all of the patients be near, at, or above a 10% dystrophin threshold. The reason that I believe that is something that is a worthy goal is that we know that in animal studies, right, a 10 to 20% level is often associated with more favorable clinical outcomes. So we know from animal models for DMD that range is something that I think the broader DMD community would also recognize is a worthy range. We know in the Becker's muscular dystrophy population that there's a wide range of dystrophin that occurs in a Becker's patient that as a percent of normal. Generally, DMD, Duchenne, is defined as 5% or less dystrophin in the muscle fibers as a percent of normal, and above that would typically be considered a Becker's patient or a milder disease that would be expected. So we know that there are Becker's patients that have milder disease with 6, 7, 8% dystrophin, but there are many Becker's patients who have more than 30% dystrophin or higher than that. And so we know there's a wide range and more variability among the Becker's population, so we really don't know how tight the correlation is to the dystrophin that's produced. The other thing we can look to is other disease areas, right? We know in the case of, let's say, some of the enzyme replacement therapies, or more recently Vertex's cystic fibrosis drug, where, again, a genetic defect that basically has these patients lacking an essential protein, much like Duchenne patients are lacking the essential protein of dystrophin. There, a value of 10% production as a percent of normal translated to very favorable clinical outcomes. So I would say I'd be looking for a consistent response that's close to that 10%. I think anything above that on a consistent manner, or any individual patients that have, something above that 15%, 20% or above, we would be very excited about. But what we're trying to achieve is a more consistent level of dystrophin that we can achieve. Now, we also know that this is only after 24 weeks, and we don't know if you can reach higher steady-state levels of dystrophin over time, which is another reason we wanted to look at that 48-week biopsy of those treated patients to see if we increase the level of dystrophin. There's always, as well, the other factor of the sampling bias, where you're taking one biopsy of a particular muscle, and we, can see variability which may not reflect the dystrophin that is throughout all of the muscle groups, or even with a particular muscle like the bicep, if you took a sample from a different part of the bicep, would you see more or less dystrophin? So it's not a foolproof measure, but the goal would be that we see a more consistent dystrophin level and something at, around, or above that 10% threshold.
Okay, great. So we have a lot of questions that have come in about specific technical things, but I first want to ask what several people have asked, and I'm not sure you can be more specific, but can you tell us when you think this press release is likely to go out?
Yeah. No, that's a big question.
Now in the AAN meeting.
I think that's really what we've communicated publicly. I'll try to provide a little more color on that. at our earnings call, we mentioned that we would have a press release or that top-line data no later than the end of April. Then we got the acceptance at AAN, and so that meant that we would have to have data to present at that meeting, right, which is April 25th. Okay. Now, I think most people understand that we're going to have some data to analyze, to review it, to prepare, to help Jerry Mendel prepare that presentation for AAN, and that we're likely to have data, sometime before that date. Okay, and so many have started speculating that could be in the first half of April, because again, it would not be in our best interest to not have enough time to review and analyze the data before it's presented at AAN. So that's the most color I can give you on timing of the press release at this time. Let me also clarify something that I heard might have been confusing about the AAN abstract. This was a very favorable thing in my mind for the company. We made a decision to submit an abstract without any data in it. And at most conferences, they would reject it outright. They wouldn't even consider accepting an abstract without any data. When we read through the AAN's guidelines on their emerging science late breaker abstract, they had a bullet point there that said only under extraordinary circumstances would we accept an abstract without data. And the subtext was it has to be a really important study, right? Knowing that we would have data from the study at the time of the meeting, for them to accept an abstract without data means that they believe this is a critically important study to understand the impact of this drug, exon skipping, on this population, Duchenne muscular dystrophy. And regardless of what the data tells us, that we think it's important to highlight and showcase at the meeting. So just to clarify, we will have data, presented at AAN. It's just that no data was in the abstract, and we thought it was very notable that the American Academy of Neurology thought that this was important enough of a study to accept, even though they weren't able to review any data prior to accepting the abstract. So I hope that clarifies that as well.
Yeah, I think that's helpful. Of course, there are a lot of questions about exons other than those you've listed here on the last slide, and if you have plans to ultimately try to address as many exons as possible that are skippable.
Yeah. So we show this slide because we think it highlights, in this case, five of the top exon skipping drugs. These five specifically comprise about 40 to 45% of all of the exon skipping amenable DMD patients. And so, as you would expect, we started with a exon skipping drug that could treat the most patients based on the number of mutation types that an exon 51 drug could treat. Exon 45 and exon 53 are the next most prevalent, so we were very pleased to announce that collaboration on exon 45. We are continuing to pursue grant opportunities around exon 53 as well, and exon 44. And so these are the ones we highlight because we think these are exon skipping drugs that it's feasible to move toward a clinical program where we could enroll enough patients to be meaningful in evaluating the safety and efficacy of these drugs. When you go beyond the top, 6 or 7 exons, it becomes very difficult to imagine doing clinical studies where you could enroll enough patients where it would be meaningful to take to the FDA to prove safety and efficacy on the other, again, you need, dozens of drugs to treat every patient that would be amenable to exon skipping technology. We believe that the FDA and the EMA understands that there's no feasible way to do clinical trials for every single exon skipping drug because of the rare nature of these exons. And we believe that there's a path toward what we have described as a class approval. That is, if we show a robust data set with our technology using our morpholino backbone, that we could find a path to get approval on other exon skipping drugs to treat rare mutations without doing the required clinical studies. But we would likely have to prove in vitro expression of the protein or maybe some genotox to show that when we change the sequence that we're targeting, that we would expect to see the same efficacy we've seen with the first drug or the second and third drug as well. It's premature to give specific guidance around this because we first need to prove this with our first lead program, and then we can have a more productive dialogue about how we could advance the program to go after these other exons and ultimately all of the exons, including the most rare. This is not dissimilar to what Vertex is trying to do with their cystic fibrosis drug platform to go after other gene mutations in cystic fibrosis. And so we are encouraged by the progressive dialogue that's been coming out of the regulatory bodies to understand in the new era of truly genetic-based personalized medicine that there has to be a mechanism to get drugs approved where you've proven safety and efficacy for certain gene types to extrapolate that to more rare gene types. So all I can give guidance on right now is that we're focused on exon 45 and 50 to follow exon 51 and to try to get an open IND by the end of 13. But it's our goal and our mission to try to help every DMD patient out there, regardless of how rare their mutation might be.
That's great. And to clarify, an IND is an investigational new drug application. That just means that AVI is able to test that drug in humans for the first time. So that's the beginning of clinical testing when Chris refers to open IND. Can you comment on whether or not you think there's any possibility we'll be able to skip multiple exons, and is that something AVI is investigating?
Yeah, I think I've described that as almost a later step in the process, right? So first, we need to demonstrate safety and efficacy of our lead program. Then we want to pursue other more prevalent exon skipping drugs so we can start to build a case for a class approval. Then the third step would be to gain a class approval. And at that point, if there was, a desire by a physician to treat a patient that would require more than one exon skipping drug, we would expect that physician to make that choice of pulling the appropriate exon skipping drugs off the pharmacy shelf to treat those types of patients. So I think again we know combination drug development can be difficult. I think a ability to use two drugs to treat a particular patient is made easier if the safety profile seems to support that you would not expect to see cumulative toxicity. But I think it's premature to expect that before we've gotten multiple drugs or a class approval for exon skipping.
Okay, so let me move on then and address what is sort of the elephant in the room, and that is that many people on the phone are aware that there was a recent decision about the patent for skipping exon 51 that was, where Prosensa's technology was given the primary significance in Europe. And I think it would be helpful if you could just talk about the fact that there are two companies, well, two together, Prosensa and GSK, and AVI, who are both developing very similar products for similar populations. I think a lot of people are worried about how that's going to play out. Is it going to impair development? Is it going to impair access in either Europe or the U.S. for these products? And I think it would be useful if you could just put that in context and also talk about what the, in the context of the orphan drug filing as well, which has some patent provisions.
Yeah, no, that's great, Sharon. So, and I talk about this frequently when I'm out with investors as well, so I'm happy to answer the question. So, we have a competitor, as you all are probably aware. Placenta and GSK also has an exon 51 drug in development and also a broader exon skipping program with follow-on exons as we do. In November, there was a hearing before the European Patent Office, which was a culmination of our opposition to a patent issued to the University of Leiden that was licensed to Placenta and ultimately GSK as the partner, which we believe was overly broad. And there were many exons named in that. There were 11 exons in total. That, again, we felt we had some arguments that thought that this was an overly broad patent. The outcome of the hearing we felt was very favorable to us in that we were able to amend claims or invalidate 9 of the 11 exons, and 9 of those were of the top, exons that in prevalence. So they were very important exons to ensure our freedom to operate in Europe. Unfortunately, we did not prevail on 2 of those 11, and those happened to be exon 46 and exon 51, our lead program. So it was a disappointment in that the lead program that we have that is showing a lot of promise to date, we would be hesitant to pursue development and commercialization based on the ruling of this patent opposition hearing. There is, a right to appeal. The written summary of that patent opposition hearing has not been posted yet. There are guidelines of when you can file an appeal. We are evaluating our appeal strategy internally until that time. To be fair, Placenta GSK, they also have the right to appeal the hearing as well. Okay, this is the unfortunate reality of the business side of drug development, where these patent issues are often outstanding. They can often be ambiguous. There can be a lot of gray area in interpretation or the probability of winning on appeal, et cetera. And many times these aren't resolved for years, if ever, and oftentimes it ends up in some kind of settlement to try to reach an agreement that includes economics of some sort. So we are intent on finding a way, if this drug is showing utility, to find a way to have this available to patients in Europe. But there's a lot of things that need to happen for that to take place. Now, one is through the appeal process, which can be lengthy and can take a lot of time before that's resolved. We are open to having a discussion and dialogue with Placenta and or GSK as well for a license of that 51, and 46 patents, so we can move forward in Europe. We just have to do everything we can to continue moving our program forward, even in light of that November patent ruling. So, again, we were disappointed of not prevailing on exon 51 and exon 46. But we are comfortable with our position in the U.S., so we continue to be optimistic of getting our drug on the market and available in the U.S. The other exons, as I mentioned, we feel we strengthened our position coming out of that November patent opposition. So hopefully that addresses the question and the concern. We will keep the market apprised of any further developments on the patent front that we see.
Okay, and I think people want to be very clear that your patent position in the U.S. for all of the exons that you're looking at is pretty solid.
Yes, we have a issued patent on our lead program, eteplirsen, in the U.S., which has an expiry in the mid-2020s with extensions that can go beyond. We're very comfortable that we are not infringing any patent out there, that we have a strong patent position in the U.S., and good freedom to operate. Yep.
Okay, I think that covers most of the questions about the patents, and I'm sure someone will chime up if there are additional ones. Someone else is wondering about non-ambulatory boys and why they have not been included in this development plan. And of course, if we think about the GSK plan, they do have non-ambulatory arms to their studies. And I wonder if you could just comment on that.
Yeah. No, we are very interested in seeing the results of our phase 2b study and understanding if it's producing the utility in these ambulatory boys that we've studied to understand how that could be extrapolated to the non-ambulatory. We have included some measures in our phase 2b that might give us an indication of how this could help and impact non-ambulatory patients, including the MBI-CTS muscle contraction test, where we're looking at the upper extremity as well, because we understand that the quality of life improvement could be significant and profound by retaining upper arm mobility and the ability to really function with the upper body. So we had to determine what was the most efficient way and best way to show the utility of our drug that would lead to the fastest path toward an approval. We know from precedent studies and populations that those markers tend to be ambulatory markers, walking tests, Gowers maneuver, forced stair climb test, etc. And again, the first point of order is to show a drug effect and to show something that would be meaningful to the FDA. We wanted to start there and then build and extrapolate that to the non-ambulatory, which again, if the data is strong, we would be intending to do that. One of the other questions that was in the research community was, is there a point where there's too much muscle damage that an exon skipping drug would have an impact, that you would be able to improve or whatever function was remaining, would you be able to maintain that function when you have a more progressive disease or a non-ambulant state? And we don't know that answer to date, but we needed to start in a population that we felt we have the best chance to show a treatment effect, and then go from there to start expanding that. We also get the question, what about younger boys, right? You're including 7-year-olds. The same thing applies, right? We don't think a 4, 5, or 6-year-old would not benefit from an exon skipping drug, but we know from other studies that they tend to still be growing. Their muscle mass is increasing. They're overcompensating for the progressive nature for the disease with the growth of their muscles, and it can confound study results. We did not want to be in a position where we would have to say we can't get the drug approved because we included a population that was broader, and then everybody in the DMD community loses out on not getting potentially an effective drug because of being too broad in our inclusion criteria and being harder to overcome some of those confounding factors. So again, we are not intending to be exclusionary in any way. We are just trying to get this drug approved in the fastest, most expedient way possible.
Okay, I think we're coming close to the end of the hour, but we've had a few questions from people trying to understand, I guess, the difference in the technology used by ProSensive GSK and that used by AVI. Just briefly, if you wanted to say just a few words about why these are not exactly the same drug.
Yeah, no, that's a great way to close. And also, it does relate to, you mentioned orphan drug exclusivity. I think anybody's interpretation of the orphan drug would suggest that these are different chemistries, different backbone chemistries. But ultimately, that's where it starts. It's the backbone of which each company is attaching the subunits, right, of a certain sequence and a length to target the specific exon, we believe is an important differentiator. And I highlight this often in my corporate presentations at investor conferences and elsewhere, where we believe our morpholino backbone chemistry is unique. It carries a neutral charge as opposed to a negative charge of the 2-OMe chemistry. And we also believe our safety profile is evidence of this. We have evaluated the placenta chemistry in their published articles, and we do see the proteinuria, the injection site reactions at relatively low doses, even lower doses than what they're studying in their clinical program. And to date, we have not seen any of those markers. We have not seen the kidney marker elevations. We've not seen injection site reactions. And so from that vantage point, we believe we have a very differentiated safety profile that we believe links directly to our chemistry, our backbone chemistry. We are also very encouraged by the reduction of inflammatory infiltrate, which suggests that we're not causing any kind of untoward immunogenicity. We haven't seen anti-dystrophin antibodies from our therapy. We're seeing a reduction in these T cell counts in the muscle. So all of that is very suggestive that this is a very safe drug. So far, it looks like we can dose this over longer duration of therapy, maybe chronically for life without seeing any long-term toxicities. We will follow these patients in this U.S. study. As long as the program stays moving forward, we would want to continue to capture safety data with that chronic administration of these 12 patients we're currently following. From an efficacy standpoint, there are articles out there that show the evidence of exon skipping at various doses. Again, at the end of the day, we believe that the ability to push our dose higher to remove any variability we might see at lower doses, to get as much exposure to the muscle as possible with our drug is an important factor. And we think that safety advantage may lead to an efficacy advantage by allowing us to push the doses much higher and to be able to dose that chronically over time to continue producing that dystrophin. So that's what I would say about how we view the differentiation of our chemistries.
All right. Thanks, Chris, very much. We're right at the end of the hour, so I want to thank you very, very much for providing this information and remind people that this webinar was recorded and will be available later. And also that if you have asked a question and you haven't answered, we can try and get to that offline. So thank you very much.
Great. Thank you, Sharon. Thank you to PPMD, and it was a pleasure to be able to address the DMD community through this forum. Thank you.
All right. And we'll conclude now. Thank you very much.
All right. Bye-bye.
Bye-bye.